Hello students, welcome back to my YouTube channel Chemistry and Biology Concepts HSTR 2022-23 about the chapter Tissue and the detailed study about the tissue. So complete chapter we are going to learn in this class. So make sure that you are making the notes for this. This will help you for the final revision of this concept tissue. Introduction to plant tissue, that is histology. It is the branch of biology which deals with the study of tissues. The word histology is used by Chat. Histology means it is a branch of a biology which deals with the study of the all types of tissues. We call it as a histology. The word histology is first used by Chat. Anatomy means the study of internal structure of various parts of a plant body. So this is about anatomy. Morphology means the study of external characteristics of an organism is called morphology. Tissue means it is defined as the group of cells which are similar in structure, origin and perform common function. Coming to the types of plant tissue. The plant tissues are broadly classified into two main types, meristematic tissue or undifferentiated tissue or embryonic tissue. Another one type is permanent tissue or differentiated tissue or matured tissue. So plant tissues are classified into two types, that is meristematic tissue and the permanent tissue. So coming to the next very important, that is meristematic tissue. Meristematic tissue or undifferentiated tissue or embryonic tissue. The term meristems, which means divisible, the cell which have a capacity of dividing itself, these kind of characteristics we can see in the meristematic tissue. Hence the name they have given meristems, that is divisible. The word meristematic tissue is given by Nageli in 1858. In the 1858, Nageli was given the meristematic tissue word. Understanding? So it is defined as a young undifferentiated cells and have a capacity of cell division and produce new cells. Coming to the general characteristics of a meristematic tissue. So the cells are equal in size, isodiametric in shape, but they may be polygonal or fusiform shape. The shape of the cell will be either it may be a isodiametric or it may be a polygonal or fusiform in shape. They are also found in the tip of a root and shoot. In the tip of a root and shoot system, we are going to identify these kind of meristematic tissues. The cells are embryonic in origin and hence the name call it as a embryonic tissue. So meristematic tissues are also called as embryonic tissue because it originates from the embryonic cell. So these cells are compactly arranged. That means compactly arrangement means where there is no in between the cells there is no intercellular space. The space in between the cells will not be there, and cells are interconnected by from one cell to another cell there is a connection. These connection we call it as a plasmodesmata. So this is about the meristematic tissue or undifferentiated tissue or embryonic tissue. So coming to the types of meristematic tissue. So depending on the origin, meristematic tissues are classified into three types. Depending on the origin where these meristematic tissue will be present, depending on that origin, these meristematic tissues are classified into three types: apical meristem, intercalary meristem and lateral meristem. Apical meristem means the name itself suggesting that it is present at the growing tip of a stem and root and also increase the length of the plant. The intercalary meristem, another one type is intercalary meristem. It is present at the base of the leaves or internodal region of the twig. That means in between the stem there is a small gap. In between those gap you are going to get internodal region. Nodal region where the leaf and fruit parts will going to be arises. So this is the internodal region. You can observe in the picture also. Intercalary meristem there they have mentioned. So intercalary meristem present at the base of the leaves. So this helps in the elongation of the plant. Coming to the another one important type is lateral meristem. These lateral meristems are present in the stem part of the plant and increases the girth of the plant. Girth nothing but the thickness of the plant will going to be increased with the help of a lateral meristem. So coming to the next another one type, depending on the origin and development, the meristems are classified into three main types, that is promeristem or primordial meristem, primary meristem and secondary meristem. Promeristem or primordial meristem means it has a group of young meristematic cells which are found in the small areas at the tip of a shoot and root. They have thin cell wall, prominent dense cytoplasm. The cell division is reduced and primary meristematic tissue will going to be arise from this primordial meristem. So coming to primary meristem, it is derived from promeristems, nothing but primordial meristem, and these cells undergoes rapid cell division. When compared to primordial meristem, primary meristems divides rapidly. Coming to the third type, that is secondary meristem. They develop from non-meristematic tissue called the permanent tissue. They loses the ability of cell division by the process called dedifferentiation. The meristems developed by differentiation of permanent tissue are called as secondary meristem. Coming to the another one type of meristematic tissue, depending on the function how they perform, these meristematic tissue again classified into three main types. First one type is protoderm, second procambium, third ground meristem. Protoderm, it is the outermost region of the meristem differentiated into epidermal tissue. So protoderm then after get differentiated and then produces a epidermal tissue of the plant. Next, procambium, it develops into a primary and vascular tissues. Primary vascular tissues arises from the procambium. These we call it as xylem and the phloem, and another one is cambium. So coming to the ground meristem. Ground meristem means it is the middle region which gives rise to fundamental tissues like hypodermis, cortex, endodermis, pericycle and the pith. So this is about the types of meristematic tissue. So here in this class we have discussed about the types of plant tissue, in that meristematic tissue and the permanent tissue. Meristematic tissues are again classified depending on the origin, depending on the development, depending on the functions. Depending on the origin, it is classified into three types: apical meristem, intercalary meristem and the lateral meristem. Depending on the development, primordial meristem, primary meristem and secondary meristem, and again depending on the function they have classified into protoderm, procambium and the ground meristem. So this is all about the types of meristematic tissue. It's about the permanent tissue. The permanent tissues are differentiated tissues. They have definite structure, size and functions. The cells of the most permanent tissues lost the cell division. That means the continuous cell division will going to be lost by the permanent tissue. They are produced by the meristems. Coming to the types of permanent tissue. Permanent tissues are again classified into two main types: simple permanent tissue and the complex permanent tissue. Today in this class we will discuss about the simple permanent tissue. So coming to the simple permanent tissues, it is composed of only one type of cells. Hence it is also called as homogeneous cells. So simple permanent tissues are again classified into three types: parenchyma, collenchyma and the sclerenchyma. So coming to the first one type, that is parenchyma cells. So parenchyma cells, it is a permanent tissue found in all groups of plants and have a specialization. Hence it is called as a fundamental tissues. It consists of living cells which are isodiametric, round, polyhedral or oval in shape. Coming to the subtypes of parenchyma cells. So these parenchyma cells and their particular subtypes, they are going to ask the functions about each cells. So one by one with the detailed study we are going to discuss here. Coming to first one, simple parenchyma. So it has an active cytoplasm and essential for the metabolism of the cell and helps in the storage and secretion. Which parenchyma cells helps in storage and secretion means, so that is simple parenchyma cells. Second one is chlorenchyma. The parenchyma cells which has a chloroplast in it, this we call it as a chlorenchyma, which carry out the photosynthesis. For these chlorenchyma cells found in the mesophyll of leaves. Coming to the third one, that is aerenchyma. In the aquatic plants you are going to get these kind of aerenchyma cells where the parenchyma cells develops more intercellular space and that intercellular space is filled with the air, which helps in the buoyancy or exchange of gases. For example, for these aerenchyma cells are that is lotus plant. Coming to the fourth one, that is stellate. The elongated cells encloses the space in banana. The parenchyma cell stores the minerals that we call it as idioblast or raphids. So coming to the another one type of a permanent, simple permanent tissue, that is collenchyma cells. So collenchyma cells means it is a simple permanent tissue consisting of living cells. The cells have a thick cell wall and deposited by a wall material called pectin. So pectin is absent in the parenchyma cells, and here in the collenchyma cells wall material is made up of pectin. The cells are compactly arranged cells without intercellular space. In the parenchyma cells intercellular space will be there, but it is absent in the collenchyma cells because of the deposition of pectin. These cells may contain chloroplast and carry out photosynthesis. Some of the collenchyma cells that present in the stem part of the plant that way will carry the photosynthesis process. Again the collenchyma cells are again classified into three types depending on the pectin deposition, that is angular collenchyma, lamellar collenchyma and the locular collenchyma. Coming to the first one, angular collenchyma. These cells are irregularly arranged and compactly arranged, and pectin deposition will be occur at the corner region. So this is about angular collenchyma. Lamellar collenchyma means the cells are with a tangential cell thickness. Tangential wall thickness means in some part of the cells it will be very thick, deposition of pectin will be very thick, and some part it will be not thick, that much thick, that is thin it is in nature. So last type that is locular collenchyma. It is irregularly arranged cells with intercellular space and appears like a hollow cylinder. So this is about locular collenchyma. So we have discussed about collenchyma cells. Coming to the third one, permanent tissue is sclerenchyma cells. It is also a simple permanent tissue consists of hard dead cells and they are in mechanical in function. So parenchyma and collenchyma cells are living cells. So sclerenchyma cells are dead cells. The cells at the young age were living with vacuolated protoplasm and nucleus. When it get matured, no, these cells are after lignification, that is lignin is the wall material that present on the cell wall. It expels out nucleus and protoplasm and become the dead cell. So these sclerenchyma cells helps in mechanical support for the plants. Again the sclerenchyma cells are classified into two types, fibers and scleroids. Coming to the fibers, they are elongated and polygonal in shape with pointed ends, originated from procambium and ground meristem. The fibers are also classified into two types, that is intraxillary fiber and the extraxillary fiber. Depending on the xylem where it will be present, if it is present inside the stellar part, this we call it as a intraxillary fiber, and if it is present in the outside, that is extraxillary fiber. So coming to the scleroids. Scleroids, these are also commonly called as sclerotic cells or stony cells. Common name for scleroids are sclerotic cells or stony cells. They are short, broad and isodiametric or irregular in shape. Again, these scleroids are classified into 5 types. So that is brachyscleroids, macroscleroids, osteoscleroids and astiscleroids and trichoscleroids or filiform. So these scleroids along with the diagram I have showed here in the picture, you can observe here. So this is about the sclerenchyma cells. Coming to the difference between parenchyma, collenchyma and sclerenchyma cells. So this is about the parenchyma. First I will discuss about the parenchyma cells. These cells are oval and isodiametric in shape. These are living cells. Presence of intercellular space. Wall material is very thin because of the absence of wall material. Cell wall is very thin. Found in the cortex and pith region of the plant. And along with that, storage and photosynthetic in function you are going to get in the parenchyma cells. So coming to the collenchyma cells means these cells are elongated and oval and living cells. Intracellular space is occupied by the pectin. Cell wall is thick because of the pectinization. Found in the hypodermis and around the vascular bundle. Vascular bundle nothing but xylem and the phloem. Living mechanical support and photosynthesis function will be done by the collenchyma cells. Last one, that is sclerenchyma cells. These cells are polygonal and tubular in shape, having a dead cells. Intercellular space is absent in the sclerenchyma cells because of the presence of a wall material called that is lignin. The cell wall is thickened by the lignification found in hypodermis and around the vascular bundle. Purely dead mechanical support will be given by the sclerenchyma cells. So this is the difference between parenchyma, collenchyma and the sclerenchyma cells. So this is about the complete concept about the simple permanent tissue along with the detailed study. Complex permanent tissue. The complex permanent tissues are composed of different types of cells, not only one kind of a cells, different types of cells will be present in the complex permanent tissue, but they are together in a specific position in the body of a plant. The position of that cells will be in a particular position, it will be there. They consist of both living as well as a dead cells because of the presence of thick cell wall. The two complex tissues are a xylem and the phloem. They together forms a vascular tissue. That means the xylem and the phloem are the vascular tissues that will forms a one particular function in the complex permanent tissue, which helps in conduction like water and minerals and as well as the food. First we will discuss about the vascular tissue, that is xylem. So coming to the xylem, it is a water conducting tissue. So xylem, nothing but another name wood. It's a water conducting tissue referred as a wood. This is composed of both living cell as well as the dead elements. They are thick walled due to the deposition of lignin. So lignin is also one of the type of a cell wall material. So coming to the xylem tissue, in that they are having a living and dead elements, I said no, as it is. The xylem tissues are classified into 4 elements or components, that is tracheids, trachea or vessels, xylem fibers and xylem parenchyma cells. We will discuss detailed study about one by one. So coming to the first one, trachea or vessels. They are cylindrical shaped, shorter tube-like cells. Their end walls are absent. End walls are absent in that and having a cylindrical shape. Shorter tube-like cells will be present. Cell wall is thick due to the lignification. They are the good conduction of elements than the tracheids. When compared to tracheids, these trachea or vessels are a good conductor of water when compared to tracheids. So coming to the second one element, that is xylem fibers. These are the sclerenchyma fibers which are associated with the xylem, provides mechanical strength. They are thick walled due to the presence of lignification, that is wall material, and it is also a dead cell. So coming to the third element, that is xylem parenchyma. When the parenchyma cells are associated with the xylem, this kind of element we call it as xylem parenchyma. They are the only living cell element of a xylem. The cells are thick walled with the living protoplasm and nucleus, which helps in the storage in function. So now coming to the fourth element, that is tracheids. They are elongated and polygonal shaped cells. Cells are with pointed ends that I have shown in the figure, you can check it out. Then they are dead thick walled cells. Protoplasm is absent and it forms a lumen in that. It conducts water and also give mechanical support due to the lignification deposition. Lignin deposition occur in the following 5 ways. One is annular, that I have shown here in the picture, you can make it a difference. That is at every intervals it is like a ring shaped. And second one is a spiral, that spiral you have observed no, that spring like adjacent rings that will joins in that. Next third one is reticulate, nothing but a network like you are going to get these lignin deposition. And scalariform means transverse interconnected bands. Here you can I observe the difference of lignification in each of the types. Next last fifth one is pitted, that is uneven type of lignin deposition you can observe. So this is about the xylem elements or components. Coming to the another one type of a complex permanent tissue, nothing but phloem tissue. The phloem tissue are also called as a blast tissue. So The xylem tissue is called as wood. Phloem tissue is also called as blast. The word phloem was coined by Nageli in the year 1858. It is a complex permanent tissue consists of as well as living and dead cells. Either thick walled cells or thin walled cells you are going to get, and also called as a food conducting tissue. As in the xylem also, the phloem is also classified into 4 elements. So, these are the elements of phloem. Sieve elements, companion cell, phloem parenchyma as well as phloem fibers. So, coming to the one by one detailed study about sieve elements. Sieve elements are nothing but they are the phloem elements composed of 2 units, that is sieve tubes and sieve plates. First, sieve tubes. It is elongated cylindrical shaped, and you can observe in the diagram also. So, these sieve tubes, it is elongated cylindrical shaped. They are living cell with a protoplasm, but nucleus is absent. Hence, it is called as enucleated cells. So, sieve tubes are having a protoplasm, but nucleus will be absent. That's why it is called as enucleated. The cell wall of a sieve tubes thick due to the deposition of a wall material called cellulose and pectin deposition, but absence of a lignin deposition here in the sieve tubes. Coming to the another element, sieve plates. Sieve element, that is nothing but sieve plate. The cytoplasm of one cell is connected with the next cell through the perforated sieve plates. It is also called as cytoplasmic strands or a transcellular strands. You can observe in the picture I have showed here. So, this is about the phloem element, first one, that is sieve elements. Coming to the next, companion cell. They are narrow, slender, specialized cells associated with the sieve tubes. These companion cells are associated with the sieve tubes. They are metabolically active cells and having a companion cells are living vacuolated protoplasm and nucleus. That means vacuolated protoplasm means presence of a vacuole in it. Next, companion cells and sieve tubes are produced by longitudinal division of a single meristematic cells. Hence, it is also called as a sister cells. It is very important. You should note down this point. In pteridophytes and gymnosperms, the companion cells are absent, but the presence of a specialized cells called albuminous cells, they perform the same function as a companion cell. They are going to ask you about which phloem tissue element having a sister cells, that you should mention it as a companion cell. As well as they are going to ask this one also, where you are going to observe albuminous cells means it is there in the pteridophytes and gymnosperms, because there will be absence of a companion cells. These albuminous cells also perform as the companion cells that you can observe in the other different types of plants. So, coming to the third element of a phloem is phloem parenchyma, and fourth one is phloem fiber or blast fibers. So, coming to the phloem parenchyma, as I have said, the phloem tissue which is associated with the parenchyma cells, we call it as a phloem parenchyma. These phloem parenchyma are living thin walled cells of parenchyma associated with the phloem. The cells have active protoplasm and nucleus. They are absent in the monocot. So, coming to the fourth phloem element, that is phloem fibers or the blast fibers. As you have learnt in the xylem fibers, these fibers nothing but sclerenchyma cells that associated with the phloem. This is the only dead cell or element you are going to get in the phloem tissue. They are sclerenchyma cells associated with the phloem. They are very thick due to the presence of a cell wall called lignification and provides mechanical support, and they are commercially very important. These fibers, phloem fibers, are very important and gives mechanical support. The vascular bundle is defined as a strands of completely arranged xylem and phloem tissue. So, these xylem and phloem tissues you have already learnt in the complex permanent tissues. So, with or without cambium, if the xylem and phloems are compactly arranged in a strand, we call it this one as a vascular bundle. So, in the arrangement, The xylem in the vascular bundles are different. Depending on that arrangement, they have again classified into two types, that is endarch or centripetal type, exarch or centrifugal type. So coming to the endarch or centripetal type. In this type of arrangement, the smaller protoxylems are towards the margin and the layer of metaxylems are towards the periphery. So the name itself suggesting endarch, inside, the protoxylems will be present at the margin and the metaxylems towards the center. In the inside that you are going to get this kind of a xylem arrangement. This is, we call it as endarch or centripetal type. And next coming to the exarch or centrifugal type. In this type of arrangement, smaller protoxylems are towards the center and larger metaxylems are towards the periphery. So coming to the difference between this endarch and exarch. So before that, you have already learnt that you should not forget about that xylem and phloem. These are the conducting vascular tissues which are arranged in a strands from the vascular bundle. So there is a two difference in between the endarch and exarch. So as usually, the endarch means protoxylem is towards the margin and in the exarch protoxylem is in center. Metaxylem is center in the endarch and metaxylem is marginal in the exarch. Centripetal development occur in the endarch type and centrifugal development you are going to get in the exarch type. So these endarch type you can found it in the root parts and exarch type you can found in the stem parts. This is the main and important difference between endarch and exarch. So coming to the next concept, types of vascular bundles. So types, there are mainly three types of vascular bundles you are going to identify here. In that, first one is radial type. Vascular bundles means you have already learnt, this is about the xylem and phloem arrangement. So radial type means in this type of vascular bundle, xylem and phloem are found in a separate bundles like a patches. So xylem and phloem shows alternate arrangement and equal in number. This type of vascular bundles we call it as a radial type of vascular bundle. So coming to the second type of vascular bundle is conjoint type of vascular bundle. Here in this type of vascular bundle, xylem and the phloems are combined and occur in a same radius. So this kind of a vascular bundle we call it as a conjoint type. In the conjoint type, again it is subclassified into conjoint collateral type, conjoint bicollateral type. So coming to the conjoint collateral type. In this type of vascular bundle, how it will be arranged means xylem and phloem remain sided by the side arrangement and occur in the same radius, that is collateral. Conjoint bicollateral means the vascular bundles of phloem and the xylem, the cambiums are two set you are going to get in the bicollateral type, but in the collateral type there is no cambium present inside that. So coming to the third type of vascular bundle is concentric type of vascular bundle. Radial, conjoint, in that conjoint, conjoint collateral, conjoint bicollateral. Now third type is concentric type. In the concentric type, a vascular bundle, if one of a vascular tissue, that is either it may be xylem or it may be phloem, that will be present in the center and another vascular tissue will be surrounded externally. This kind of vascular tissues bundles we call it as a concentric type of a vascular bundle. Again this concentric type of vascular bundle is again classified into two main subtypes, that is amphicribal or hadrocentric. Another one is amphivasal or leptocentric. So coming to the amphicribal, here what happens, in this type of xylem occur in the center and the phloem surrounds the xylem externally. So this type of vascular bundle we call it as a amphicribal or hadrocentric. So coming to the next subtype, that is amphivasal or leptocentric. In this type what happens, exactly opposite to the amphicribal, that is phloem present in the center and xylem surrounds the phloem externally. So coming to the another one very important concept, that is annular ring. In the spring wood and summer wood together we forms a 1 year growth. These 1 year growth we call it as a annular ring. So these annular rings helps in the finding the age of that particular plant or a tree. So this is how it will going to form means with the help of spring wood and the summer wood, that we will discuss later. So coming to that, dendrochronology, the branch of biology which deals with the study of annular rings. This kind of a branch we call it as a dendrochronology, very important. And the xylem produced in the summer, the xylem when it will produced in the summer or autumn season, this is and are less in number and they are narrow and thicker. The activity of the cambium is differentiated by some seasons. The cambium is more active in the spring season and it is unfavorable in the summer season. As I previously said about the annular ring, in that heartwood and sapwood detailed study we are going to learn here. So heartwood are also called as a duramen. So the central dark brown colored hard wood give mechanical support to the plant. It gradually loses the conduction ability or capacity. Then they become filled with the oils, gums and tannins. This kind of wood we call it as a heartwood or duramen. Coming to another one wood is sapwood or alburnum. Here sapwood or alburnum means the marginal light yellow colored region consisting of young xylem. They are compactly soft, hence it is called as sapwood. Here these heartwood and sapwood will form a annular ring. So coming to the difference between heartwood or a duramen and sapwood or a alburnum. So it consists in this one, what happens, the heartwood means it consists of secondary xylem, but in the sapwood it consisting of younger xylem, because it is present at the peripheral region and it is very light yellow in color, but heartwood will be dark brown in color. And the lumen, that the space present in between that, the lumen is blocked by tannin and tyloses. Tyloses I will give the definition in the next slide, so you can go through that. So these lumens are blocked by tannin and tyloses, but in the sapwood what happens, lumen is not blocked because it is consisting of a young xylems which helps in conduction of water. Because of this heartwood, it provides a mechanical support because it is having a secondary xylem. Sapwood helps in the conduction of water and minerals. So this is about heartwood and sapwood. As I have said about the tyloses, in the xylem element, the balloon like growth developed into their lumen. So these type we call it as a tyloses. It is very important for HSTR exam. They are growth of a parenchyma into the lumen of a xylem element, that you should remember. So parenchyma cells will going to be grow in the lumen of the xylem elements in the form of balloon like structure. The development of the tyloses convert sapwood into heartwood. This is about the tyloses. The study of wood is a separate branch called silviculture. Very important, you have to remember. So this completes the chapter tissue and the detailed study about the tissue. I hope you guys understood the concept very well. Please like, comment and share. So please make sure that you have subscribed to my channel. Thanks for watching.